Right. So we don't have a podcast interview from 10 years ago.
We should.
Because we would have asked them to make a prediction. And now we could test whether it came true.
We should check that.
Prediction. I don't know, whatever you said 10 years ago when we interviewed you.
Exactly.
We can now find out.
Well, I will look it up when.
Well, if we not look it up now, is it?
No, probably it's.
We can. It should be on the CS network.
The internet should be able to find it for us.
I have the lecture here.
No.
So
Oh,
our
oh
first
my
speaker
God.
today, it's a real pleasure to introduce.
Cool.
11 years ago.
Yeah, it was 2nd of September, 2014.
Exactly.
And my first slide was a Misty Oxford, and Krubisa told me you should apply for grads in asthma, not in neuroscience.
And did it work? So looking here for
Google's not helping.
What favorite question?
An interview. Maybe we can do one. We do one for everybody.
I think so. 2014, right? So let's. Here you can go to the archive. This is 18.
But you already had robots, I remember.
Oh yeah.
You showed me robots.
Mm-hmm.
Then you showed me the walk-in neural circuits. I remember that.
Mm-hmm.
So you were probably way ahead of your time.
But nobody cared. No. Okay, so let me see what I have here. Do we have? We have these maybe cortical organization. I don't see development really as a separate topic, Tony.
Shall I look at my talk?
Devo.
Oh, right. Actually, we talked about reptilian mammalian transition, how neurogenesis is cranking up. I think some of the progenitors were discovered.
Here you are.
And
this is the Convergent Science Network podcast.
that's Anna.
Leading researchers in the domain of neuroscience, brain theory, and technology are interviewed by Paul Verschure and Tony Prescott.
It's Paul Verschure, together with Tony Prescott of the Convergent Science Network podcast of the BCBT Summer School. And today we're here with our speaker, Zoltan Molnar.
Okay.
And Zoltan was speaking about the evolution and the development of the neocortex.
I scroll to the end and see what the prediction.
Yeah.
Really? You
the cortex.
We have that.
So I'm fascinated.
He's fascinated.
Yeah.
So the last 3
The
minutes.
cortex will remain there. So I'm fascinated with the association of these scattered residual cells
to
cognitive abnormalities. So it's like when you have sloppy builders, they leave some of the scaffold behind where the adult structures cognitive abnormalities. So it's like when you have sloppy builders, they leave some of the scaffold behind when the adult structure is finished. Probably they haven't done a good job anyway in the adult structure because they left the scaffold inside and then they can't remove it. So I'm fascinated with these, and that's why I'm so interested in development, because I I agree with Willis from 1664 that many of the cognitive
I'm very good.
disorders, they are brain developmental abnormalities, and they are really
They will say
important.
stop the fucking
And these
TV.
transient scaffold cells, or the remnants of them, they tell us quite a bit about how the brain is constructed. And simply because these are the earliest generated cells, to understand them and understand their evolutionary origin, that's why I do quite a bit of comparative work. So although it's a bit more clinically driven, what I want to emphasize is that sometimes it's good to just step back and look at the bigger overall picture. And although it's a simplification, but the subplate is maybe the reptilian framework of our mammalian brain.
And then, if we're going to meet up with you in Oxford 4 years from now,
4 years.
yeah, we're going to remind you of this discussion of today. And we're going to tell you, look, 4 years ago you made this prediction, and now we're going to go check, see what came out. What's this one prediction you would like to make you feel most passionate about today in your work?
So if in the next 4 years I could monitor and modulate these transient scaffold cells in the mammalian brain and show that by modulating their function could alter cognitive development, then I would be very, very happy. And if I could extend it some neuropathology, so histology or even imaging, we have 7 Tesla machines, and if I could show that abnormal cortical development will have an impact on these subplate cells or vice versa, and then I would be very happy.
Okay, great. Zoltan Molnar, thank you very much for this conversation.
Thank you, Paul, and thank you.
A little bit unspecific.
Yeah, he's sort of a bit wishy-washy there, right? Okay, so, well, we got work to do now. Are you up for this, Sultan? Are you ready?
Yeah. Are you?
No, we're recording. We're ready to go. We're
Even the sorry, Leah, about the comments of the.
So okay, we're back here in 2025 with the Convergent Science Network podcast, and it's still Tony Prescott and myself, and we're back again with Zoltan Molnar, who was speaking at our ACBT school on neurodevelopment. And it's also interesting, of course, that more than 10 years ago, we also had an interview. He also participated in our summer school then. And one of the things you started out with as an overview in your overview statement was that the brain development is prolonged, right? So it's a continuous process. And also you drew this strong link to evolution and development, right? So when you say prolonged, so what do you exactly mean with prolonged? The prolongation, how long?
So if we compare the human brain development, the proportions of the times allocated to setting up connections, existence of the transient projections with the consolidating adult connectivity, it's extremely long. So we have a lot of time to self-organize based on the input we receive. And I think some of this is not present in some of the models we use today. Mm-hmm. So some of the murine models, some of these steps that, for instance, thalamic projections arrive to the cortex and then they accumulate and then they start interacting with the forming cortical circuits. These are within hours or days maximum. Whereas in human, will go on for several months during in utero life. And also it will continue within the first postnatal years. And I think we are just not used to it, that we have this long period of coexistence of the transient circuits with the adult circuits. So I think we will probably need better models to study these transient circuits.
But does that also imply, well, two things. First, how do we measure time in this case? Mm-hmm. Is it like relative time? That means relative to life expectancy, right? And then maybe the relationship might be different, right? Because what's the life expectancy of a mouse and also their developmental time? So let's say, what,
2 years
30,
max.
40% of the time they're in development. Humans might, if we take like a life expectancy 70 or something like this, you also talk about 40, 50% of that time is in a developmental phase in some sense. But so if you look at life expectancy of the organism, isn't that not becoming constant?
I think Barbara Finley established a website called Translating Time.
Mhm.
And I think there are all sorts of species where they have the data on certain steps of development they lined up. And I think you can predict, what is the relative maturity of a newborn ferret to a newborn mouse. But the question you asked, I don't think anybody ever looked at.
But wouldn't it be a
To see the
reasonable-
developmental time in relation to the life
like for
expectancy.
instance, brain volume scaling with body size, right? So maybe developmental time scales with life expectancy, right? And then we have an invariant relationship. And then maybe it's a surprise, that's not a big surprise.
It would be very interesting. Good question. Yeah.
But if you had to make a bet, what would be your bet? Would it be constant, or do you expect that there is a difference between, let's say, rodents and primates?
So, looking at this Translating Time website of Barbara Finley and colleagues, I wouldn't make any bets because we assume that that the development is universal and you can compare the time. No, there are some structures where you have a faster development and evolution is speeding things up, and maybe the cortical development is actually slower. So, for instance, a marsupial, a Mododelphis domestica, when it's born, it's like embryonic day 11 mouse, but the brainstem is extremely mature and the spinal cord, because they have to make it all the way from the uterus to the nipples, because otherwise they don't get food. So with their brainstem, they crawl up, pop onto the nipples, and then they start developing further. And then their cortex will develop a bit later. So the brainstem is maturing much earlier than the cerebral cortex in these animals. So I wouldn't be surprised if some subcortical brain structures would develop with a different speed compared to the cortex.
So I think you answered the question almost in your talk, because chimpanzees, you said their brain is mature in 2 years.
Well, the size, yeah.
So but the life expectancy is 30 plus years.
Yeah, but that's size, right? That's not, let's say, white matter tracks and connectivity and so on.
Neuropil and the thickness of the cortex and these kind of things. It plateaus at 2, whereas we plateau at 11, 12.
Yeah.
So, and our living expectancy is only double or triple. Yeah. So.
Okay.
So maybe, but I will look into this because it would be interesting too.
Yeah, but because the second implication then is, if you say it is prolongated, that then raises the question, is it prolongated as some self-organizing process, or is it prolongated by virtue of having some meta-controllers that ensure that it develops as a stable system? Right? Because imagine it's just a self-organizing process. The more time you give it, the higher the risk that it will become a bit stable. Right? So does it suggest that there is a meta-level developmental controller at work?
I think what is probably happening is that it's not a homogeneous maturation across the cortex. So I think some cortical areas crystallize way before some others. So for instance, the frontal regions, they don't myelinate until you are 21 or even later. So probably there is a reason why we leave it so late. And also there is a sequence how these connections develop. So for instance, the thalamic projections get to the proximity of the cortex very early on. They accumulate in the subplate before they make their final move eventually to the ultimate target cells. So, but in certain areas, this happens much earlier than than in others.
But the prolongations, of course, so it suggests there's a finish and a start, right?
Exactly. And also then the connectivity back from the cortex to the thalamus is even more complicated. The first descending pathways are from subplate, but they are overtaken by layer 5 projections. And in fact, layer 5 is the first cell type which is entering the thalamus. The subplate and the layer 6 projections, they somehow slow down and they accumulate outside the thalamus. So they are in the proximity, but they don't quite enter the thalamus. And in the mouse, although they were there at embryonic day 15, they only enter, for instance, the natrogenic nucleus about postnatal day 10. Can you imagine how long it is that they wait outside before they go in? You enucleate the animal or you block the activity of the retina, then they move in immediately. So there's something interesting, some regulations going on.
How do you interpret that?
I don't know how to interpret it, but basically the activity pattern within the thalamus has an impact on the kinetics, how these cortical thalamic loops close. And maybe there is a reason why you close these loops.
Well, what I was worried about with respect to thalamus is that, of course, these recurrent projections have to target both specific or non-specific nuclei and the reticular nucleus, right? So you have to do both.
Yeah.
And this sounds to me like a really tricky problem to solve, right? Because you don't only have to worry about the feed-forward projection, because once you hit the reticular nucleus, you really start to have divergent inhibitory effects, which you could imagine could destabilize this whole. So would that be a reason to not innervate fully, that you first want to have this feed-forward pathway converge in some way in order not to destabilize? Is that a way to think about it?
Actually, you put your finger exactly. This is the thalamic reticular nucleus is the suspected location of the weighting of these corticofibular projections. So unfortunately, nobody did, including us. We haven't done a really nice, reliable tracing on this. We used transgenic animals where we had enhanced green fluorescent protein expressed in the subset of these cells. And you can see that the green fibers accumulate outside the thalamus and they wait there, probably at the site of the thalamic reticular nucleus, and then they move in. What is also interesting, they do have functional connections with the thalamic reticular nucleus. About more than 20 years ago, I stimulated the cortex and looked at the voltage-sensitive dye recordings. What do the corticothalamic projections do close to the thalamus? And they do drive the thalamic reticular cells at very early stages, probably when they are not even inhibitory yet. So there are all sorts of transient circuits, not only the subplate and the thalamocortical projections, but also the corticofugules, thalamic reticular nucleus, and then the thalamus. The first person who suggested that you have these circuits was Karlashatz and Paschko Rakic. They did some proline, old-fashioned proline undergrad tracing from the cortex in the developing macaque, and they noticed that the radioactivity was waiting outside the thalamus before they moved in. So we did a bit more work on the rodent, and it's clear that something funny is going on outside the thalamus. So these loops don't get closed. And I would go even further. If you want to understand these cognitive abnormalities where you have these loops, it's not enough just to look at the subplate. You also have to look at what's going on outside the thalamus. And it was, I think, Ray Guillory who first suggested that these thalamic reticular cells, they were also much more numerous during development, and they are kind of like the subplate of the thalamus. They shrink, and so it would be a very good idea to have a look at the relative numbers in cognitive disorders, just as it was done for the subplate.
Okay. So are you saying that what's happening in the cortex is maybe not unique to cortex, but there's a similar developmental mechanisms going on in other structures?
So for the thalamocortical projection, the subplate is the transient zone where they accumulate and then they move in. For the corticofugules, it's maybe the thalamic reticular nucleus. So that's kind of like the subplate of the thalamus for these fibers. So there are several transient cell populations, and the thalamic reticular, perireticular nuclei, they are the other transient.
But if we were to look elsewhere in the brain, would we see things like subplates structuring the growth of the migration of the neurons?
So I think in the spinal cord they describe the border neurons. They are also transient, and they were attributed some developmental role, but we don't know whether in certain pathologies you have more or less cells remaining. So thalamic reticular, nobody looked at it in cognitive disorders, and I think it would be very...
And say supercaliculus, which is another laminar structure.
Right.
Is there anything for the...
I'm not aware of any transient layers in the thalamic reticular nucleus, but it would be good to do some proper 3D systematic birth dating and then look at what are the hotspots, which cell groups disappear in large numbers. And even the presence and absence of the preferential cell death in rodent subplate is debated still, because we don't use the proper entire brain reconstruction. So we don't agree what percentage of these cells disappear. Some would say that they all stay. Juan de Carlos and Laura Mascaraque, they would suggest that most of them stay. And Chiaki Oktakamariama in Tokyo, they would say that most of these cells will stay. Whereas I would say there is preferential cell death between postnatal day 2 to 8, and this is when some populations decline in number and others, the relative proportions increase. And we, about 15 years ago, we pulled out markers which label these more permanent cells.
But how can you have a disagreement on that? Because that seems to be a very objective measurement.
It's very objective.
So how can you disagree?
Because I think the reason why we disagree is because even subplate cells, they have subpopulations. So you have some subplate cells which are born at embryonic day 10, others 10.5, 11, etc. And I think maybe the early born cells behave differently from the later born subplates.
So with the classification issue.
It is, and also subtypes. And what would be really good is to have markers for the early, late generated, permanent, non-permanent cells. Right. But we are getting there.
What Tony was suggesting could be an interesting way to think about the forebrain development, right? So you have a bunch of subplates that you position, and then from there you grow the substructure, right? So it could be a way to think about how neurogenesis plays out for the forebrain. But then you were saying, well, it's not so clear whether it's actually true for any other structure but the cortex, right? So what's your hypothesis? What direction does it go in? Because imagine we say, oh, cortex is an exception, right? This doesn't hold for all these other structures. They still have to be developed in some way.
So these structures are the precocious, very early generated cells, and they form a continuous layer which is providing a supporting scaffold on which the rest of the cortex will develop. So I don't think that these cells really interfere with neurogenesis, but they do interfere with regulation of migration and also stabilization of later connections. So you have a zone where you have relatively mature cells. They can already communicate with other cells. They set up some connectivity, even at an early stage when some of the neurons are not generated yet, not in position, and also the connections are not functional. So they receive some transient connectivity. They have the waves, activity. These are the first regions of the brain where you have synapses. And then when the cortex is finished, then the connectivity is moving on. So the thalamic fibers move into the cortex, cortical goes into the thalamus, and then these transient cell populations in subplate and in thalamic reticular nucleus, they reduce. And many of them will disappear. So I think this process could be inhibited by altering the development. So if you remove sensory input, or you block cell death, maybe you would end up with a very different circuit.
Mm-hmm. So then, on the other hand, what you mentioned in your talk is that from the subplate, not all neurons disappear, correct? But there's a number of neurons that remain that are distributed, if I understand it correctly, relatively evenly across the whole cortical sheet, playing a very fundamental regulatory function. Right. So how should we call these cells? How are they embedded in the cortical circuits in the adult brain? How should we think of them also in that control function?
So I was also very surprised to see that nature is using these cells for these fundamental functions, because I believe that these cells are extremely important in regulating local arousal, attention, perhaps local sleep, and why leave it to a cell population which is very important during development, but only have a couple of scattered cells in the white matter, or a sublayer, one or two cell layer thick sublayer in layer 6B. So, but it looks like that these cells are the one which are really fundamental. So evolution is repurposing these cells in the adult.
But morphologically, what do they look like?
They are just like any other neurons. You have multiple morphology, multipolar. Some of them, they have transient apical dendrites, which is reaching layer 1 during development. And then at certain stages, these projections neurons, they retract this apical tuft, which is quite common for other cell populations as well. You also have bipolar, multipolar, neurogliaform. You can have glutamatergic and GABAergic subplate cells. Some of the early GABAergic subplate cells have very long-range connectivity, which is also very interesting because maybe they have some kind of a hub role.
So they're very heterogeneous.
Very heterogeneous, and they come from all sorts of different locations, and we don't know which cell type is the one which is causing perhaps the cognitive dysfunctions when you have more surviving. Those studies were done very early with relatively simple markers like NADPH, diaphorase, and
But that implies, Zoltan, that maybe these neurons are not even homogeneous system.
Yeah.
It might be also the subsystems, right?
Correct. Yeah.
Okay. And where do the exons go?
So it depends on which cell type. So for instance, we have a couple of Cre lines where you have in mouse where you have you think that you have a subpopulation of these cells, like DRD1A Cre cells in the subplate. Maybe it's like 15% of the subplate cells or layer 6B cells, and yet we've found out that they have 4 different types within that DRD1A Cre, which is only the 15% of the subplate. So even if you have a genetically defined subplate population, so it's getting a bit complicated because you can only monitor or modulate these cells where you have control over them. And even this DRD1A Cre line, which is quite selective, is heterogeneous. Then you have the CTGF Cre, with connective tissue growth factor Cre expressing, and that is slightly different population. So you ask me, where are the projections? We studied both of these. The DRD1A Cre is very interesting because they have projections up to layer 5 and also up to marginal zone. But they also have projections out, subcerebral projections to the higher order thalamus, which is selective to the higher order thalamus, and also some to the superior colliculus. And they have actually quite wide range connectivity within the cortex. And interestingly, the other cell types, the CTGF Cre, they don't have this widespread and no selective connectivity. So the DRD1A Cre cells, it turns out that they are exactly the cell type which is orexin sensitive. The CTGF, they are not. So that's why we study especially the DRD1A Cre, because I think they have a very strong grasp on these cortical, thalamocortical pathways, and they regulate the activity. And that's why we believe that they have a role in arousal, attention, and maybe even sleep.
Mm-hmm. Okay, but wouldn't one hypothesis be, because these are all subplate neurons or cells,
Or derived.
so they can take on multiple functions early on in development. So couldn't you interpret them as sort of a default solution? Like, okay, I have a little cortical circuit. The cortical circuit is generating various gradient signaling, some pieces missing. And now these progenitor cells turn into the function that that little subcircuit requires. You see what I'm saying? It's like filling in gaps, filling in developmental gaps in the circuit.
So you cannot produce more, but you can maybe regulate their survival.
Exactly.
What you mean.
Yeah, they have a circuit, but it is not fully functional. So the gradients are that are being generated in the circuit indicates well, we miss cell type X, and now the progenitor neuron turns itself into cell type X in order to just make sure this column or this little subcircuit can survive.
Yeah, so I completely agree, but again, you use the term that progenitor turns into. No, these cells are already there, but they don't disappear. So exactly.
Okay,
Yeah, you know the distinction. So basically, I completely agree with this. So you can envisage that these cognitive disorder, the conditions, they have more subplate cells because the subplate is implicated. But you can also imagine a situation that the cortical plate is altered, it's sick, it cannot develop, therefore you need more of them.
Right. Exactly.
And this is not the primary cause, it's kind of a so we don't know exactly. So what would be interesting is to manipulate some of the cortical cells and have a look what kind of impact they have on the survival of the subplate cells. So
but it's interesting because it's a hypothesis that would be orthogonal to yours because it puts
the cortex
the cause at the cortical level and not at some subcortical projection somewhere doing something.
Okay, right, but the outcome is the same. You have more. You have more subplate cells, and they have this sensitivity and the abnormal connectivity, which will then have an impact on the cognitive function. But I just wanted to tell you that there is evidence for this, some kind of correlation. So with Bashir Ahmed, who is retired but then joined my lab to do some projects, he looked at macaque cortex and correlated. Well, his question was very simple. We have variations in the interstitial white matter cells in sulci and gyri. And he wanted to know what cell type would follow this variation in the subplate remnant cells in the cortex. And layer 5 and the proportions of layer 5 and number of layer 5 cells is very strongly correlating with the proportions of the subplate cells. So here we go. So there is a matching between 5 and 6B or interstitial white matter cells. So maybe we have to look for the cause in 5.
Yeah.
So there is a possibility.
It would be a more minimal interpretation, right?
Right, but
there's a functional impact.
But on the other hand, we also looked at some of the susceptibility genes of autism and schizophrenia, and we looked at where are these genes expressed in the developing cortex. And the strongest affiliation was the subplate. So that would argue that actually the subplate is the primary cause of the abnormalities.
But then in the autism case, so morphologically in terms of cortical circuits, it's this ratio of short and long range projections that is affected, right? So that there's a reduction of long range interactions. I mean, it's one of the anatomical signatures, if I'm correct, right? So then, if we look at the subplate neurons that remain, how could they have such an impact on the cortical morphology or connectivity? Or would you see this as independent, unrelated? Like, or to take any other pathology you looked at, right? So how could they influence on these other signatures that we might find in cortical circuits. Well, what's cause and what's effect here? Right?
So if you have an abnormally high density of these cells, or you change their distribution, I can explain why you have a cognitive dysfunction, because these cells feed into layer 5 marginal zone, and also you have lots of cortical connections. So if you have more of these cells, then that could argue for some of these cognitive dysfunctions. The other way around, when you are saying that you have a damaged developing cortex and therefore you have more cells surviving, that could be also explained with the connectivity, but we don't know where the primary cause was. So whatever the case, it's a good idea to study these cells.
In your talk, you showed various examples, right?
Right. But we don't know what was the cause.
Okay, so that's correlational at this stage.
So Akbarian looked a lot of schizophrenia cases, and indeed there was always elevated level of these interstitial white matter cells, and especially in the prefrontal areas. And I also showed some studies for the autism, exactly the same, that you have more cells. But the cause, it was not really explained.
Okay.
So in mouse, it would be very easy to do some experiments. I would manipulate some of the cortical plate cells and have a look whether I have more interstitial white matter cells left. So I think we looked at layer 5 silenced brains, where genetically I silenced layer 5 by removing SNAP25 member of the snare complex, but I didn't see any change in the subplate in these. Layer 6B, but maybe we didn't look hard enough or not the right area, so I wouldn't bury that still.
But you were saying that these subplate cells are responsive to orexin.
Correct. And
so does that suggest, I think you're suggesting some sort of attentional type function for these circuits. But I mean, is that a hypothesis about what they're doing when they're initially there? So they arrive, they network with each other, possibly with other parts of the brain, and already in the very early child, they have some function before the rest of the cortex has developed. But we don't know what that function is, but can we speculate that it might be to do with arousal or attention? I mean, what would you suggest?
I think it's probably it's kicking in a bit later. I think these neuromodulatory functions. I don't know what is the earliest time when they have orexin sensitivity. We did not study that. What we did study was that if you silence these cells genetically and then you apply orexin, the orexin has a different effect on the cortex. So the orexin effect is mediated through these through this subpopulation of layer 6B neurons, and that's why it's such an interesting potential target for therapy, because you could alter anxiety without affecting the rest of the brain.
And why do you think that's later developing? Why wouldn't that be?
Because I think the major target of these orexinergic fibers is the prefrontal cortex. So I don't know when it is, what is the earliest time when applying orexin would have an effect on the prefrontal. Okay. So I think we could check it.
But we know that before the rest of the cortical microcircuit is assembled, these neurons are already present, already networked, already active. They're doing something.
But these are mostly for glutamate and the thalamic input. So I don't know when they become sensitive to all the neuromodulators.
So we don't know if they're sensitive to sensory input.
To sensory input for sure. They are very early on. So Patrick Kennel showed it in ferret, and ferrets develop very early. So a newborn ferret would be extremely immature. And Patrick showed that auditory signals are mediated through subplate in the newborn ferret. Without the cortical plate in the auditory cortex fully developed. So the subplate is the first cell group which will mediate the sensory input, whether this is proper sensory input sensory-driven activity or just spontaneous activity in the receptor.
Right, but they have the connectivity.
They have the connectivity
that they could be
to stimulate
responsive
the
to,
subplate. Yeah.
I mean, for example, if we would look at visual, say B1 in the newborn infant, so the subplate cells will be there at birth. They'll have some network.
Even before.
They'll have some sensitive. Well, the reason I picked B1 is that there isn't any visual input before the birth.
Right.
So you open your eyes, there's light. Now, you already have a circuit here that is ready to respond to something.
Correct.
But have we any idea what that response is?
So I think the visual cortex by that time is almost fully wired up. And you remember there was a huge debate whether ocular dominance columns are ready in the primate before birth or not. And in fact, I think the result of the debate, but I don't read the recent literature on this, but I think it was that in primate, the ocular dominance columns are there, but to stabilize them, you need coherent visual input later on. So it is surprising that the spontaneous activity can sort things out like this.
Right.
So, but subplate is playing a very important role in utero life, setting up all these connections. So in the auditory cortex, for sure, they have a very important role, and also in developing ocular dominance columns, they have a role. And also, I showed some experiments from Patrick Canod and Kai Kaila when they, in the rat, they destroyed selectively the subplate cells, and they showed that no barrows developed in the barrel cortex. So, so all these three modalities, it has been shown that subplate has an important role.
But Zoltan, to go back to Tony's point about orexin, why is that significant? Because orexin as a peptide can have all sorts of effects, and I think we don't really understand the full portfolio of impact it can have, right? It can, in principle, again, modulate how other modulators work and so on, right? So why is this orexin observation then really telling us something about how the circuit operates? I'm just trying to understand, right? I'm confused at this stage.
So your brain is wired up, and in different contexts, you have to activate different parts of your brain. So you behave very different when you have no sensory conflict. You think you understand everything, and also when you have some kind of a unusual stimulus present. So I believe that this orexin system has a role in activating the system, which is very important in resolving these conflicts, and you have unusual stimulus. So this arousal is waking up these higher order cortico-thalamo-cortical circuits, and you begin to pay more attention to that stimulus until you resolve it. So the reason why we became interested in orexin, because there is a very strong selectivity in the responsiveness of these cells. So it has been known for a while from a group in Geneva, I think more than 20 years, that these projection neurons in layer 6B are the only projection neurons which have orexin sensitivity. Now it turns out that the orexin sensitive ones are the ones which communicate with the layer 5 and marginal zone and the higher order thalamus. So they are perfectly positioned to wake up the whole system. Now, the very same neurons, they also have response to neurotensin and dopamine. So somehow they are kind of a neuromodulatory hubs. So they are, in number, they are very low, but you only need one conductor in an orchestra, no? And then
but would that be sufficient? Because you could say, look, they impacted the sleep-wake cycle very slow, by the slow process. Or you could say, So that they set the overall operating mode. Like, are we awake or are we asleep? Or you could say it's attentional, but if it's attentional, it's actually a pretty fast system. And there we have other neuromodulators doing things, right? You could think about noradrenaline, or your locus coeruleus, you have the raphe nucleus, there's a forebrain acetylcholine. Lots of them have sort of attentional effects, right? So do you see it in this fast system or in the slow system, or both?
Both. And I think it could be in many different ways. As you know, for instance, if we just talk about orexin, so you have about less than 10,000 cells in your lateral hypothalamus. Then they go everywhere in the brain, subcerebral, and also in the cortex. In the cortex, they go mostly to the frontal part of the cortex. When you fall asleep, the front and the back of the brain, they have different velocity how you fall asleep. It's not a universal thing. So that's why I became really interested in the possibility that these remnant cells of the subplate can have this very important adult function. And the other beauty of this whole observation is that finally we can link the developmental abnormality with the circuit which has the dysfunction in the adult.
So with how many of these subplate neurons, these remaining subplate neurons, that have orexin as or excrete orexin as a neurotransmitter or peptide?
They don't excrete, they respond. All the orexin is produced in the lateral hypothalamus, and then it's flooding the brain.
So diffusion,
it can be both. It can be synaptic release, and also it can be more like endocrine release. And it's interesting that if you look at the orexin GFP mouse, where all the fibers are green, which have orexin, they don't respect 6B. They go everywhere in the cortex. But the receptors on the projection neurons only in 6B. There are some interneurons which respond to orexin, but for projection neurons, these are exclusively just in 6B. So I was so surprised to see then that I always consider the hypothalamus as a ancient swamp of neuromodulators which are occasionally released, and they look after your very ancient functions, the eating or mating or these kind of things. But it seems that these lateral hypothalamic neuromodulators, they know what they are doing. They target very key structures which regulate the state of your brain to process higher cognitive functions. So probably we had this hypothalamic system even when we were in the reptilian stage, and we started evolving a huge telencephalic vesicle, and some of the receptors for these hypothalamic, they are strategically positioned to take advantage of this brain state regulation.
Yeah. So, but then, if it's this hypothalamic regulation, is it setting an operating mode, or can it act very fast?
I think this is a very important question. So we don't know.
Okay.
I don't know. I predict that you will have two types, a bit slower and a direct. The reason why I'm saying that there is a direct, because you can do a Cree-dependent rabies tracing. So if you want to know which cells talk to these subplate remnant cells, you can express Cree recombinase. You inject a rabies receptor, which will be expressed only in these starter cells. Then a few weeks later, you inject your rabies virus and you detect which cells talk directly to these layer 6B cells. So Tim Zornig in Berlin and us in Oxford, we looked at this in different starter cell populations. And the DRD1A Cree frontal cortical subplate remnant 6B cells, they receive direct lateral hypothalamic input from the orexinergic cells.
Right.
So that's why I believe that probably it can be fast and also slow, both. So So these lateral hypothalamic cells, they have the potential to directly talk to these 6B cells.
But then you also mentioned that they modulate the thalamocortical interactions, right? So and that was actually another part in your talk that you emphasized quite a bit.
Right.
This sort of the role of the thalamocortical projections as if you want a backbone of a lot of the also more advanced cognitive functions of the cortex, right? So how should I look at this interaction between these hypothalamic driven systems and the thalamocortical one? Do you see them as competing? Are they synergistic? What's their relationship?
So basically, I think they are quite separate in a way that imagine that you have this cortical thalamocortical system. So the layer 5, layer 6 projections, they have very selective connectivity into the higher order thalamus. So the part of the thalamus which is not receiving direct sensory input, like the pulvinar. And the orexin system is selectively targeting this part of the thalamus and the input cells which project to that part of the thalamus. So I think the orexin system is selectively waking up this cortical thalamocortical via the 6B.
Mm-hmm.
And that could probably open up these channels to resolve things which we don't find clear answer immediately. So, for instance, if I would predict that if you use the so-called oddball paradigm, that you suddenly change a very predictable stimulus, and then this system would wake up, and we don't have evidence for that, but this is what we would like to study.
Yeah.
But we do have evidence that if you inactivate these layer 6B cells, then you actually reduce anxiety in the animals.
Yeah, but so you also mentioned that the thalamocortical system was sort of structured in a hierarchical fashion, right? That was one other point that you made. And you saw sort of a bifurcation in that. Let's say we have simple functions and we have complex functions. So then, and these must also then be modulated in some way. So now you're saying the orexin system is more, let's say, non-specific wake-up call to that system, but also to switch it into a mode of, let's say, problem solving. So is that the right way to think about it?
Right.
Yeah.
So,
but then you're saying, sorry to interrupt, but then you're saying that the hypothalamus says, we got a problem that I don't know how to solve. Right?
Yeah. So, so how to show this directly? Because this looks like a very nice hypothesis. Yeah. So I think it can now be resolved by pushing down some really long electrodes which penetrate all these structures. So not only through the cortex and through layer 6B, but then you don't stop. You keep going through the first order and higher order thalamic nuclei, and perhaps even, and then record the activity pattern after you confirm the position of certain electrodes. You show when they are really active in what behavioral situation. So in vivo awake behaving with these electrodes in the strategic location could resolve this. So I would predict that when you have some state transitions from, a bit dormant, predictable stimuli into something unexpected, that these 6B cells and higher order thalamic nuclei will be much more active.
Okay.
That's my prediction, but I have no evidence so far.
Yeah, but isn't it a bit counterintuitive? Because you could also argue arousal means there's an unpredicted event. That means also very primitive behavioral systems start to kick in, because now defense comes in, freezing might come in, or defensive attack might come in. And in all these behavioral systems, in some sense, also are effective because they essentially shut down cortical processing for a little bit, because it will take too long. It's slow. It has to integrate. It has to deliberate, right? So I got to act, has to survive. Right? So isn't it a bit counterintuitive to link an arousal linked system to actually a state switch in the cortex that will move it towards the phase of deliberation, which makes you vulnerable.
Yeah, but that's why we are human. We have a huge higher order autonomic system. That's why we can resolve things, and you still have to activate this system for that. So this theory of attention and arousal and through this system is not excluding all the other possibilities which we might have. For me, it was surprising to see just how specific the receptor distribution is of some of these neuromodulators. So there is a reason why this layer 6B subplate remnant is a neuromodulatory hub. And it turns out that even psychedelics act on this cell group preferentially. So they can also open up channels for all sorts of associations when you apply psychedelics. So layer 5 and layer 6B have the most interesting receptor distribution. So it cannot be a coincidence. There has to be some functional explanation.
Yeah, but to me, more switching in the opposite direction of what you're describing. For me, it's more like, let's shut down the frontal cortical part because that's just a risk. Right. Let's get the hell out of here. Let's freeze. Okay.
Right.
So that's my counter hypothesis.
Right. Okay, but when you are in an unusual environment, then you need to switch this on. But you don't want to switch it on too long and too strong, because then you might have anxiety. So that is, all these states can be actually continued. No, arousal, attention, arousal.
Of course, it's leave it. Right?
Yeah.
Sure, but for me, an arousal system also brings you back more into a primitive behavioral control because it's non-specific, and if it's non-specific, I don't know what to do, so I better fall back on non-stereotype behavior. If it's specific, it means there's no arousal. I can just commit myself to the test. I'm not under threat. Now I can deliberate. I can weigh my options and so on. Right. So this is why I was. To me, it's counterintuitive, therefore, to link that system to actually deliberation.
Well, time will tell how these interact.
Of course.
We select, but
sure.
At the moment we just have this.
But what do we know behaviorally, like in animal studies? If you inhibit this orexin system or you stimulate the orexin system, what's the behavioral result?
So, interestingly, orexin got its name from Professor Yanagisawa, because he believed that orexin has something to do with appetite and food intake. It turns out that probably not. Orexin was causing arousal in these animals, and therefore they were eating more at that time.
Okay.
So it's more of a sustained arousal which orexin is doing. Now, orexin has probably another 50 or 100 targets in addition to the cortex. So it's very difficult to kind of study the function of the orexin selective to cortex. But in our case, when we silenced the recipient cells of this orexin signal to the cortex, it had a strong anxiolytic effect. And that's what we would like to study a bit more. Our study was not ideal because we silenced the entire cortical mantle, and also the population we selected, the DRD1A3 cells, is not a clear cell population because you have some contamination of other structures. But now we can actually target subregions of this and have a look whether we can actually alter anxiety. So that's one of the research projects we are doing right now.
Right. So, but then, Zoltan, earlier we listened to the prediction you made more than 10 years ago, and the answer was a bit aspirational, no? So now, we're 10 years further down the road, and of course you gained a lot of experience and understanding in the meantime. So if we're now going to get back to you in Oxford 3 years from now, what's the specific hypothesis you want to see validated in 3 years' time? What's the specific one that is a do or die? That's the whole of your career going down the tubes if you fail, right? So what's the one that you really want to see tested?
So when we met last time, we were talking about this reptilian framework of the developing mammalian brain, and I think what became clear is that yes, there are some cells which have this very ancient origin, but it also became clear that in human you continue to produce these cells. So it's not just an early generated cell group. You continually add. So a couple of weeks ago, Tom Novakovsky, UCSF, published that in human, the short progenitors, they produce subplate cells later, and they get added to this archaic cell population. So it seems like if you have a bigger brain, you need a bigger scaffold, and therefore you keep producing it longer. So that was a very interesting discovery recently. So we still maintain that they have evolutionary origin, but there is in human, at least you have a later generated cell a population. Now, 10 years ago when we talked, I was dreaming about showing these modulatory functions of the residual subplate cells, and I think we delivered on that.
Yeah.
So
Yep,
if we chronically manipulate this cell group, then we can actually change arousal, anxiety, and sleep. We didn't believe that this was possible, but that's and the byproduct of that study is that we also manipulated layer 5 cells. And with Vlad Vyazovsky, we discovered that if you manipulate layer 5, then you have a strong manipulation of sleep. So 10, 20 years ago, the dominant idea was that sleep is bottom up. So all the subcerebral structures like hypothalamus, brainstem, they regulate sleep and they tell the cortex when to go to sleep. Now we know that the cortex has a big say in when it's going to sleep. So, and it's layer 5 and layer 6B, which is actually regulating this, and perhaps some other interneuron populations. So that was also a big breakthrough. And now what I would love to do the next couple of years is to find a way to selectively manipulate the number of these cell groups and then have a look at the behavior of the animal. So one possibility is to change the mTOR pathway, and we have been doing it with Britta Einkort and Matthew Larkham at Charité in Berlin. And what we want to do is to manipulate these early generated cells and their preferential cell death and have an animal model where you have more cells. And actually, I think we accomplished that because we now have a model where you have about 30% more layer 6B cells. And now we are looking at the behavior. So it will be very interesting to see.
What's your prediction on behavior?
So I think you will have to use probably very sensitive behavioral tests, novelty preference, or very or arousal or attention driven tests. So these are not easy to set up in mouse. So I believe that this will show some alterations in these animals. So
would you predict improved performance in, let's say, a complex maze, or compromised performance?
For the contrary, I expect that they will be a bit more timid, or they will have a bit more anxiety, more shy, and probably they will have very subtle alterations. So it will be interesting to see whether these are the endophenotypes of autism or schizophrenia, which we have. And perhaps you can even study hallucinations or even delusions in these animals. There are some methods how to do that in mice. So that would be very interesting. But my dream is to actually to have human markers for some of the cells which matter the most, to have a look at human cases and identify, which you can do now. With spatial transcriptomics, you can pull out the cell types from the interstitial white matter and have a look which cell type is the most vulnerable in these cognitive conditions.
Right.
Yeah. And perhaps there are several types of schizophrenia and also...
Exactly. Yeah. But then, Zoltan, last question. Zoltan's law, right? So you're now in this business for a long time. You're active in one of the leading universities in the world, running one of the leading labs in neurodevelopment. So if younger researchers would like to follow your trajectory, what is Zoltan's law that they should follow?
No, I think it's probably it's very exceptional that somebody is sticking to one question. Most people change field very frequently, and that's probably good for you to change. But I'm just fascinated with this problem, and I think this problem is a very good entry point to learn quite a bit about the brain. So I might start with this cell group, but look at my research. It had to go to all sorts of other areas because of this cell group, and unfortunately, you have to learn. So for instance, I was not very keen on doing behavior, but now we have to do it. I was not familiar with local or global sleep, but now I have collaborations going on, and also I was not doing spatial transcriptomics, but I have to do it to get to the answers. So although I'm asking the similar questions, but I think for the progress, I had to change my research quite a bit.
Fantastic. Well, Zoltan Molnar, thank you very much for this conversation.
Thank you very much, Paul, and looking forward to seeing you in the next 10 years.
Exactly. You will. Wonderful. Well, thank you. No, I learned a lot from this. Very good. But it's indeed, it's amazing, right? How this all came together in this 10-year period.
I will go back and listen to
It's astonishing, right?
recording and see what we predicted and where are we right now.
But I think that there's an interesting discussion to be had here about, because also you see that your behavioral prediction, these are non-specific effects, right? It's arousal,
but
risk
we know
averse.
the circuits. No,
I
think
no.
There are some suggestions, but we don't know
Well,
what is the circuit of attention or arousal, or we don't even know the regulation of global or local sleep. So,
well, no, that's actually a really important point, which perfectly matches my theory of consciousness, because the whole point I'm making is that the thalamocortical system, is also just selectively switching off parts of its circuits, putting them to sleep so they go in low frequency burst mode, because that's how you also create the content that you are dealing with, right? Is by sort of dynamically switching off subcircuits. And that fits in your thalamocortical story rather well, I think.
But then the 6B is really well positioned to bring the subliminal to liminal.
Exactly.
Yeah.
By switching off the competition, and then this is how that becomes the dominant scene. And so if we sleep, we are misunderstanding the thalamocortical part of sleep. I think it's just a generic feature of managing cortical dynamics by switching things on and off. It's just in case of sleep, it's in a more global switch off state. But during wakefulness, exactly the same thing happens. If as soon as you hit the bursting mode of the thalamic relay neurons, okay, you put that circuit to sleep. It cannot process anything. It's off. And to me, this is like the sleep spindles. It's essentially the same mechanism. And consciousness is floating in that dynamic state.
Very similar. Your theory is very similar to Matthew Larkin's, who is a kind of a consciousness expert. Yeah.
Mm-hmm. Right.
And that's why he is taking 6B very seriously. Because it might be layer 5 producing all these bursts and things, but it's 6B which is telling it when and how to do it.
Exactly. So and I could imagine that your subplate neurons might be much more integrated with that part of the story, right? So that modulation. Anyway, so there's a lot of stuff to discuss there. So really,
but thanks. It's I think very interesting to think about these bigger issues. Yeah.
Well, that's what.
But life
Right,
is short, so we have to get on with it. Yeah.
Well, the thing is to build a bridge, you know.
Mhm.
So anyway, that was great. So it's great that you're still here tomorrow.
Yeah.
So we can still chat a bit, and then you stay longer here in Alicante, or you go back?
No, we go back tomorrow.
Oh, you have too much stuff going on.
Yeah.
Yes.
We have the medical admissions now, so we have like 60 students, half an hour each. But it's an important process.
Of course, no.
Because you have to select the next generation, and also it's not an easy course, so you want to make sure that you select people who are outstanding.
Yeah.
Yes.
But do you see a shift in the demographics of these students?
We still have pretty good, but I mean, to be a junior doctor in